Hi everyone. Do you see my screen?
Yes.
Okay.
Yes. Yes.
Yes, ma'am.
So where are we?
Yes, ma'am.
Thank you. Where are we?
There.
Hmm? No, I already talked about DNA replication. That I remember.
I think it starts on the telomerase.
I didn't talk about telomerase.
No, you did. I think it's the last one that you did.
Oh, so I did this part.
Yes.
And did I ask you to do the math?
No, I don't remember that part.
Oh, I have already answered. Okay, so let's start from here. So Clostridium tetani, it has 14% guanine. What percent of DNA is adenine? So if it has 14% guanine, then what's the percentage of adenine? Okay. So how are you going to do the math? So you know that every time adenine goes with thymine, cytosine goes with guanine, right? So how many percentage of adenine is there? Exact same percentage of thymine there. Same way, how many cytosine is there? Same amount of guanine is there. So over here we see 14% guanine. 14% guanine mean it has 14% cytosine also, right? So together, cytosine and guanine, it gives me 14 plus 14 means 28%. Now, if cytosine and guanine together has 28%, then we have 100 minus 20 means 72% adenine plus thymine. So that means we have 36% of adenine and 36% of thymine. So basically what I did, I divided 72 with 2 and that gives me 36, and that's the percentage of either adenine and thymine. So right answer is cytosine. Okay, so our next step is transcription. Now, transcription means where a cell make a RNA, which one is the DNA copy of, that's the exact copy of DNA actually. So, transcription for So for transcription, sorry, yeah, okay. So what we do, cell actually make a messenger RNA copy of a DNA. So let's say it's your DNA, and if this is the sequence like adenine, let's say cytosine, guanine, thymine, adenine, cytosine, guanine, thymine. If this is the sequence of DNA, then cell make a complementary RNA strand. Okay? So in this case, this is called messenger RNA or mRNA. Now, in this case, it is a complementary strand. So if the DNA strand goes from 5 prime to 3 prime, then this messenger RNA must go from 3 prime to 5 prime. And in this case, the sequence should be, this is RNA, so uracil will replace thymine. we must have then uracil, guanine, cytosine, adenine, uracil, guanine, cytosine, adenine. Okay. Now, what happened? DNA every time, and this process occurs inside the nucleus. DNA is very, very precious, okay, and carry all the hereditary information. So we really don't want DNA to come out of the nucleus because we really want to preserve all the hereditary information. So only solution is to make one mRNA copy or RNA copy. Now that messenger RNA comes out of the nucleus and it enters inside the cytoplasm. And inside the cytoplasm, we have a ribosomal subunit. Now that messenger RNA sits on the ribosomal subunit. So this is our messenger RNA. Okay, and then ribosome and tRNA together read the coding sequence on messenger RNA, and then by that way they make protein. Okay, so this part that occurs inside the nucleus means when cell make one RNA copy of DNA, that process is called transcription. Now, transcription, what happens? Like DNA replication, when we studied last week DNA replication, you have seen the entire length of DNA has been copied. Okay, there is small portion at a time, but the entire length of the DNA has been copied. But for the transcription, only the portion we need, means let's say at this moment my blood sugar level is really high. So blood sugar level really high means I need insulin hormone to control that blood sugar level. So a cell needs to make insulin hormone, that's the protein. Okay, so at this moment only the portion of my DNA that is carrying that insulin gene, maybe from here to here, that portion is going to transcribed. Okay, not the entire length of DNA is going to be copied. Now what happens? So if this is the portion of the gene we need to transcribe now, we really do not have any origin of transcription or anything. But what happens? We have a promoter region and we have a terminator region. So promoter region, that's the starting point for the transcription. So we start at the promoter region and we end at terminator region. Every time the direction goes from 5' to 3'. Okay, so basically the DNA has to be opposite, so 3' to 5'. In this case, we have special enzyme who catalyze the reaction, and the name of the enzyme is RNA polymerase. Remember, we are making RNA copy. So over here, it is not DNA polymerase, it is RNA polymerase because we are making RNA. Okay, so now RNA polymerase, it actually at the beginning, it sits at the promoter region of the DNA. Okay, and then it is RNA polymerase. Remember, it does not need any primer to start the reaction, unlike DNA polymerase. So over here, what it does, that RNA, so this is the RNA polymerase. This enzyme, RNA polymerase, just read the nucleotide sequences on the DNA, and it pick up all the RNA nucleotide. This time we are making RNA, so RNA polymerase pick up RNA nucleotide. So basically, it picks up adenine, guanine, cytosine, and uracil. So based on the sequence or based on the arrangement of the nucleotides on DNA, RNA polymerase read and pick up the RNA nucleotide and make a complementary RNA segment. Okay, and it started working from the promoter region, and then ultimately when RNA polymerase reach at the terminator region, it stop reading the DNA sequence, or you can say it stop the transcribed the DNA. So that's the whole scenario for the transcription. We have three stages. We have initiation. Initiation when RNA polymerase sits on the promoter region. Then our next stage is elongation. Elongation means when RNA polymerase synthesize the complementary RNA strand. So it reads and pick up all the RNA nucleotide from the cytonucleus, and it make one complementary RNA strand. That's the elongation part. Termination, when RNA polymerase reach at the terminator region, then the whole process is going to stop, and that stage is the termination. Okay, so that's the everything about the transcription. It's very easy. So if you understood the DNA replication last week, then this part is very easy. So you have to remember, first thing is, in this case, the main enzyme that catalyze the reaction is RNA polymerase. Transcription occurs inside the nucleus, and it starts at the promoter region. Then, and when RNA polymerase sits at promoter region, that's the initiation. Now RNA polymerase read, plus it pick up all the RNA nucleotide, and it makes a complementary RNA strand, and this stage is called elongation. Termination is what, when RNA polymerase reach at the terminator region. Every time we make, during transcription, we make complementary RNA strand, and it is complementary of our DNA. So in this case, DNA actually, the portion of DNA we are going to copy, that portion of the DNA work as a template strand. The difference, the entire DNA is not going to transcribed, only a portion of the gene we need, that portion is going to transcribe. Next thing, that DNA transcription goes from 5' to 3' direction. That means the DNA template goes from 3' to 5', because these two are complementary to each other. So that's all about the transcription. Now, as I have told, that messenger RNA, so the transcription occurs inside the nucleus. Now that messenger RNA comes out of the nucleus. Okay. Now, remember, inside a chromosome or inside a DNA, we have both coding sequence and non-coding sequence. Okay, so maybe that insulin gene, it starts from here and then goes all the way up to here, maybe maybe here. So we have another coding sequence. So if we have to transcribe that insulin gene, we have two coding sequences and one non-coding sequences. Now, when RNA polymerase make one DNA copy, we do not have any ways to avoid not to transcribe the non-coding portion. Okay, so basically RNA polymerase actually copy the entire length of the DNA, both with coding and non-coding sequence. Then that cell, what it does, it actually cuts down that non-coding portion from the coding portion of the messenger RNA, and that's how it actually saves the ATP. You can understand, like when the protein synthesis is going on, for the protein synthesis to make one protein, we cost a high amount of ATP. Okay, and it takes time. Okay, in emergency situation, if cell has to read all these non-coding sequence, and that we really do not need, that requires time plus ATP. To avoid that, what cell does, so if we have three regions, so this is coding, this is non-coding, and this is again coding, and we need both these two regions, cells actually cuts out this portion from the coding sequence. Okay, so first it separates the whole segment into three segments, and then it actually add both the coding sequence together. So ultimately we will have two coding sequence, and that's the final messenger RNA that will goes out of the nucleus for protein synthesis. Now this process where cell keeps only the coding sequence on messenger RNA. So basically the messenger RNA is very neat and clean looking. Okay, it does not have any extra burden on it. So this process is called RNA splicing. Okay, now introns means interrupting sequence, means non-coding portion, that is called introns. Okay, means it is interrupting sequence. Coding sequence means who are actually going to be expressed. So this coding portion is called exon. So during RNA splicing, what cell does, it removes the introns, all non-coding sequence, and joins all the exons, means coding sequence, to produce a continuous coding sequence. And this process is called RNA splicing. So for eukaryotic chromosome, that RNA splicing, we really do not see it. For bacterial transcription, means it occurs only for eukaryotic chromosome. Now, another thing happened immediately after eukaryotic transcription. is, so if once that RNA splicing is done, so let's say we have only all the exon or the coding sequences. So this is messenger RNA. So at this moment, messenger RNA, and this is 5' to 3', messenger RNA is ready to come out of
Auntie,
the.
can you repeat that just one more time? My phone went out and I couldn't hear you.
Which part?
Marjorie.
Oh, okay. So what happens? Let me go back. So once immediately after transcription, so during transcription, what we do? We make one messenger RNA.
Right. I got all the way into RNA splicing, but I didn't hear what you said after that.
Oh, okay. So basically what happens, so after RNA splicing means it takes out all the non-coding sequence of the messenger RNA, okay? And non-coding sequence means it is called introns because they are interrupting sequence. And coding sequence are called exon, means they are going to be expressed. Okay, so all the introns means intervening sequence actually are going to be removed from the messenger RNA, newly formed messenger RNA, and all the exons are going to be joined together to make a new messenger RNA. Okay. Now, once this messenger RNA is ready to come out of the nucleus, remember that protein synthesis occurs inside the cytoplasm, and transcription occurs inside the nucleus because DNA always remains inside the nucleus. Okay, so when this messenger RNA is ready, for eukaryotic chromosome. So this RNA splicing does not occur for the prokaryotic chromosome. It occurs only inside eukaryotic chromosome. Okay, now for eukaryotic chromosome, when this messenger RNA is ready, another thing happens. What cell does, so at this moment you can understand that only thing we have all our coding sequence from starting from the 5' end to all the way 3' end, all are very, very important to make a specific protein. So each and every nucleotide is important over here. So we really need to protect everything starting from the first sequence to the last sequence. So what actually cell does, it add a head at the with actually the first nucleotide, and it add a tail at the end of the last nucleotide. Now for the tail, it adds multiple adenine base. So that is why it is called poly A tail, because multiple adenine nucleotide base are there. So this is poly A tail, and we add one cap. This is called cap. Now for the cap, what happens? We add with adenine, we add a methyl group. So basically adenine with one CH3 group. So that CH3 group work as a cap, and multiple adenine work as a tail. So when that messenger RNA is ready to come out of the nucleus, before that, for eukaryotic chromosome, cell add one cap and one tail. Okay, and these two steps RNA splicing, and so RNA splicing and addition of cap and tail is special procedure for eukaryotic chromosome, and it is called RNA processing that occurs only for eukaryotic chromosome. That whatever I told, that exons are going to join and introns are removed. So messenger RNA is ready to go out, and then we add head cap, 5' cap, and 3' tail. Now the genetic code. Genetic code actually it shows the relationship between and messenger RNA codon and its corresponding amino acid. So remember last week I told you that every time when cell read the DNA sequence, it goes by the 3 nucleotide at a time. So 3 nucleotide sequence on DNA called triplet. On messenger RNA, this 3 nucleotide base pair, let's say this is adenine, cytosine, guanine. This is maybe uracil, uracil, uracil. Maybe uracil, guanine, and adenine. This 3 base pair, any 3 base pair is called codon. Cell actually read 3 base pair together, so it can only read the codon. It does not read individual nucleotides. It reads codon. And inside the genetic code, we have a meaning for each codon. Okay, so over here you can see this is the DNA strand, and 3 base pair, 1, 2, 3 makes 1 codon. So over here, when UGG makes 1 codon, it makes tryptosine amino acid. When 3 uracil makes 1 codon, that means cell needs to make phenylalanine. When it is GGC, then cell needs to make glycine amino acid. So inside the genetic code, for each codon has a specific meaning. So the relationship between this codon and the amino acid is called the genetic code, or you can say the representation of the relationship between the codon and the amino acid is the genetic code. Now this is the picture of genetic code. I hope you remember that we have 20 amino acids already disposed inside our body. So we have 20 different amino acids, okay? And what happened every time 3 base together makes codon. And a genetic code always goes with RNA. So inside the genetic code, you cannot see thymine, but instead you are going to see uracil. Okay. Okay. So we have 4 nitrogenous base pair, right? We have adenine, guanine, cytosine, and uracil for RNA. So 4 base pair, and codon carry 3 base pair. So this combination, if you do permutation combination, then we have 4 into 4 into 4, basically 4 codons are there inside the genetic code. Okay, so you can see this is our first base. How are you going to read the genetic code? So this is our first base. All 4 nucleotides are there. This is our second base with the 4 nucleotides. This is at our third base. For the third base, we have 4 areas, and each area carry all 4 nitrogenous base. Now, how are you going to read? Let's say our new codon is, let's say U. That's our codon, okay? So our first this is our first base, this is our second base, this is our third base. The first base, U. So our spot is over here, U. Second, U. So U, and third base U. So that's our third base. So this one and this one. So U, 3 U together make phenylalanine. You can see that. So out of 64 codons, there are 61 codons for that has specific assigned amino acid. There are 3 codons that do not have any meaning, and they are called stop codons. So we have 3 stop codon, and they are UAA, University of Alabama, University of Augusta, and University of Georgia. You have to memorize these 3 stop codon, okay? So it's, for me, it's very easier to remember that University of Alabama, University of Augusta, and University of Georgia, these 3 are stop codon. So for these 3, we really do not have any amino acid assigned. So that is why they are the stop codon. Now we have one start codon, that is methionine. This is our start codon. It is Augusta, AUG.
One thing, so just to, because my audio went out, the 3 codons.
3 stop codon.
That has no meaning. Right?
Has no meaning and has no assigned amino acid.
Is AAG?
Now UAA, UAG, and UGA. And I usually read like that: University of Alabama, University of Augusta, and University of Georgia. So that's how I can. It's very easy for me to remember. Did you get it?
Yeah, I got it.
Okay.
They all have 3 amino acid base in them, or 3 nucleotide in them.
Yeah, because that's how cell read, because that's the codon. Remember that genetic code means the relationship between codon and amino acids. So inside a genetic code, we are going to look for a codon and amino acid. So, and codon means, every time codon means 3 nitrogenous base. Get it?
Yeah, but we have 3 nitrogenous base for each of them. It's
I cannot hear you, Fizzy. Yeah, tell me, what did you say?
But since we have 3 nitrogenous base in them, and like what you say, University of Alabama, University of Augusta, and University of Georgia. But when we look at the other one, they each has 3 nitrogenous base on them. Why do they call them stop codon, or is it because
why they are stop codon? Because they do not represent, they do not have any meaning. Means they are not assigned for any amino acid. Okay. And when a cell makes protein, these are the codon on messenger RNA. When if one of these stop codon is there on messenger RNA, then cell stop making protein, and that is why they are called stop codon. And we have 3 stop codon. We have one start codon, that is AUG, but that start codon actually represent one amino acids, and that is methionine or MET. Okay, so these are 4 very important codon that you have to memorize. I can ask you, what is the stop codon? What is the start codon? Okay, now you can see that there are few amino acid who has more than one codon. Okay, like proline over here, you can see. For proline, we have 1, 2, 3, 4. 4 codons are assigned for proline. Any one of the codon should be there on messenger RNA to make that proline. Okay. For phenylalanine, we have just 2 codon. So these codons are very specific for amino acid. Like when one codon codes for serotonin, like let's say UCU codes for serotonin, that UCU will not code for any other amino acids. So they are very, very specific. So a genetic code is redundant because with more than one codon for some amino acids. Unambiguous, that any codon for one amino acid does not code for any other amino acids, and they're universal, starting from the human to all the way to the bacteria. All the organisms on earth, even the plants, we actually follow the same genetic code. So we can say that genetic code is universal. So do you have any question from genetic code? Anybody? No?
In the exam, will you give us that board if you?
In the exam, I am going to give you this genetic code. Okay.
Miss Sandy, can you repeat the part of the 4 times 4 times 4 equals 64 codons? You said that there's 4 that are nitrogen based for something and 3 nitrogen based for something else. Can you repeat that part?
Yeah, 4 into 4 into 4 means we have 4 nitrogenous base, right? We have adenine, guanine, cytosine, uracil. So 4 nitrogenous bases are there, right?
Mm-hmm.
And we have codon that carry 3 nitrogenous base. Yes?
Okay.
Okay, now by arranging these 3 nitrogenous base, but you have 4 together, you can actually make, means by doing permutation and combination, you can make ultimately, or you can say at the highest, 64 codons.
Okay, got it.
I want to say that arrangement, like you can arrange A, or you can arrange ACA, or you can arrange CAA. Do you see? Or
Yes,
you
ma'am.
can arrange AAC. Just with 2, we can make 4, or maybe more.
Yes,
Okay.
gotcha. Yes,
So
ma'am.
yeah, so actually we can get at the max 64 codons. We have 64 codons over here. Now out of 64, 61 codons has meaning, 3 codons, they do not have any meaning, so they are the stop codon. And who are the stop codon? University of Alabama, University of Augusta, and University of Georgia. Who is the start codon? It is the city of Augusta. And the start codon, it has a specific meaning. It code for amino acid mate or methionine. It is methionine. Are you clear? Can I move? Okay.
Yes.
Okay.
Yes.
Now, our next part is the translation. Translation means when finally we are going to make the protein. So what happened? First, we replicated DNA. Oh gosh. Replicated the DNA, right? That's the replication of DNA. Just immediately before cell division. Now we make a RNA copy of one DNA. Now that RNA and this thing occurs inside the nucleus. Okay. Now RNA comes out of the nucleus. Now it's inside the cytoplasm. Okay. Over here, we are going to read all the sequence and cell is going to make protein. So this is the replication part. This part is the transcription. And this part is called translation. So now we will work on the protein synthesis or the translation part. So for protein synthesis, we have few important components. Of course, the one component is messenger RNA, okay, because that's our main source of information. So cell is going to read all the information over here. Now, who is going to read? We have ribosome, and then we have tRNA, who are going to read all the codon sequences over here. So ribosomes are made up with rRNA plus protein. It has two units. One is large unit and another one is small subunit. For prokaryotes, the total weight of these two ribosomes is 70s. For us, for eukaryote, the total weight is 80s. Now, for prokaryotes, 70s means large subunit is 50s and a small subunit is 30s. Do not try to add. It's just the weight. That S represent Svedberg unit. Okay. For eukaryotes, we have 80s. Large one is 60. Small one is 40s subunit. Now, before translation means immediately before the protein synthesis, these two subunits usually remain separate, and not all the time we can even see the ribosome. Okay, immediately before protein synthesis we can see the ribosome. Now these two subunit comes together and they get attached with each other and they make a whole ribosomal component. tRNA or transfer RNA, what happens? So let me see. Okay, so this is the structure of transfer RNA. So tRNA, they actually transfer something. So what they transfer? They transfer one amino acid they transfer. Now, what happens on the head of the tRNA? They carry one special amino acid. Okay, and at the tail part, they carry the codon for that amino acid. Okay, so we have three nitrogenous base over here. Okay, now that's a codon that code for this amino acid. But when we see that codon on tRNA, we called that one as anticodon. Okay, so over here, you can see this is the tRNA. So over here, it has anticodon, and on top, it actually carry that amino acid. Now, why it is anticodon? So we find a codon on messenger RNA, right? So if this is the messenger RNA, then these three are codon. What happened that let's say this is the tRNA. The tRNA actually carry one amino acid. This is the codon for that amino acid. Over here we have anticodon, so that has to match with this codon. If these two matches, so this is anticodon, so anticodon and codon has to match. If this matches, basically this is the complementary of the codon. So let's say over here you can see the codon is GCC, then anticodon should be complementary. So for G it should be C, for C it should be G, C, G. So this is the anticodon. If anticodon codon becomes complementary, that means they match. So the tRNA can sit on the messenger RNA. If it can sit on the messenger RNA, then it can release that amino acid. Okay, and that's how cell actually make protein synthesis. So this is the tRNA, this is the amino acid attachment site on head, and this is the anticodon attachment site on tail of a tRNA. So the translation, what happens, how actually cell make protein. So this is the picture of ribosome. You can see this is the large subunit, this is the small subunit of ribosome. So what happened on the ribosome, it has two sides. One side is called A side and this is the P side. So let's say at the beginning, this is a ribosome. Okay, and this side is A, this is P, and this is actually E. E means exit. Now, I cannot change the color. Oh, I can. Okay, so let's say this is the messenger RNA. Okay. Now it is carrying the hereditary information. Okay, so this messenger RNA is going to make my black hair. To make that hair black, it requires specific arrangement of codon sequence. So codon A, codon maybe codon 1, codon 2, codon maybe 64 should be there. So if to make the black hair, if this is the sequence of codon like 1, 2, and then 64, and that is actually written over here. So ribosome is going to read this sequence. Okay, it will read the sequence, and all the tRNA, they are going to carry that specific amino acids. Okay, this is the tRNA, and then first ribosome is going to read, then tRNA will bring that specific codon over here, okay, and then ribosome will add sequentially one after another, and that's how it is going to make a polypeptide chain, and that is the protein. Okay, that's the whole scenario. Now how it happens? So messenger RNA sits over here. So it must have start codon sequence. So let's say over here it has one sequence, then this is the AUG, then it has GGC, then it has UAG. That's the sequence on messenger RNA. Okay, and these 4 codon together represent one protein. So first messenger RNA will sit at A region. So this is the A side. Once it sits over here, then ribosome will try to read. So ribosome will start protein synthesis from the start codon, and our start codon is AUG. So it will not read this codon. Okay, so ribosome will allow messenger RNA to move. So this codon does not have any meaning. Now over here, that AUG will sit. Once ribosome will read AUG, our start codon, it will send information to the transfer RNA. Now all the transfer RNA, remember we have 20 different amino acids. So all the transfer RNA, they are carrying different amino acids. So maybe amino acid 2, amino acid 3, amino acid 4. So all 20 different tRNA are there carrying specific amino acid. And at the tail, they have corresponding anticodon. So all these tRNA, they are moving, they actually try to take chance, to take seat over here. But ribosome will not allow to sit all of them. It will only allow that tRNA who has anticodon that goes with this codon. Okay, so who has UAC anticodon means who is carrying methionine amino acid. Only that one will come over here and it will sit. So at head it has methionine and at bottom it has UAC anticodon. Now these two are complementary to each other. Once these two are attached together, codon, anticodon, then messenger RNA will move on P side. So now that tRNA with that methionine, it comes over here. When it comes over here, now the next codon sit on A side. Okay, now our next codon is AUG. So another tRNA who has EUA, oh, that was the, okay, so another methionine. Okay, so it will come over here. Now when once that messenger RNA reach at P site, it will release that amino acid. Then it will move towards slowly. Basically, the messenger RNA moves from A site to P site to E site. Before it moves to the E site, means exit, this is the exit, it actually releases the amino acid. Okay, so now the second amino acid come and attached with the first one. Then third amino acid come and it attached with the second one. And that's how cell actually make a polypeptide chain. So that's the whole protein synthesis scenario.
Could you explain how? Okay, so the AUG, that's always going to be the start codon.
Always going to be the start codon. Always. If so, let's say this is a messenger RNA, okay, and it has so many different types of codon over here. Ribosome will only start protein synthesis when it will found AUG. So maybe this portion, it does not make any sense. So a cell or ribosome is not going to transcribe this part of the messenger RNA.
Okay, and then once it finds the AUG, the codon that makes sense, right? Then the complementary codon, which we call MET, right? That's when.
No, this is the tRNA. This green part is the tRNA. So, and this is the anticodon. tRNA, it has two characteristics for tRNA. On head, it has one amino acid, and tail, it has anticodon. And that anticodon needs to match with the codon on messenger RNA. Okay, so for start codon, every time the tRNA is going to carry that methionine. So that's our first amino acid. For protein synthesis, every time we start with methionine amino acid, because the methionine is methionine actually represents the start codon.
Okay.
So are you clear? Now, so basically messenger RNA moves from this side to this side, and it moves from A to P and then to E side. Okay. Now what happened ultimately when that stop codon, like let's say stop codon UGA, University of Georgia. So when that stop codon, this is one of the stop codon. Now when the stop codon are reach at the A site, then ribosome is going to stop the protein synthesis and it will release the polypeptide chain inside the cell. Okay, so that's the translation. Now for the translation, we have three steps: initiation, elongation, and the termination. Initiation means when that start codon sit on the A site. Elongation means when that messenger RNA move from A to P side, tRNA brings corresponding anticodon and the amino acids on the A side, then they move together on the P side. Once that messenger RNA and tRNA combination reaches at the P side, they release the amino acid. Okay, and ultimately when that stop codon comes at the A site of the ribosome, then the translation actually stops. So that part is the termination.
So, Ms. Nandi, every part of the process is like dictated by the A site.
Yes.
Like
Yeah, it starts from the A side. E side means this is actually the reading site.
A side.
You get it? So when which codon sits on A side, ribosome can read that codon. Ribosome and tRNA, they can read. So this is the reading side. So this is the reading side, and this side, the P side, is actually the site where our tRNA is going to release that amino acid.
And the E side, does that actually exist?
Exit means when codons are over there, that means they already have been read by the ribosome and the tRNA.
Okay.
So that's the whole process termination translation. Now the last part is the mutations. Okay, mutation means changes in the genetic code. So change in change inside the nucleotide sequence. Any sequence, change in the any sequence, like let's say inside the messenger RNA. So this is your messenger RNA, and remember the protein synthesis start from the start codon, and the start codon is AUG. So let's say I need insulin at this moment. My body needs insulin, and cell is going to make insulin. So messenger RNA is ready. It sits on the ribosome. Everything is fine. But what happened because of the mutation? I really do not have this EU. I have AAG. What will happen? Cell cannot start protein synthesis. Okay, and my body cannot make any insulin hormone. So that's the mutation, changes in the nucleotide sequence.
I'm sorry, could you say that again? It says if
like our start codon, to start the protein synthesis, every time this is our start codon, right? AUG, that's our start codon. Remember,
right.
Protein synthesis, we must have it actually start from the start codon, and AUG represent amino acid methionine. Now, because of the mutation, instead of AUG, if it is AAG. So on mRNA, if it is AAG, there is no start codon on mRNA. mRNA cannot read, cannot start protein synthesis because there is no start codon, right? So cell cannot make that protein. Cell will not be able to make that specific protein because of the lack of the start codon. So what happens over here? There is a mutation. Just only one nucleotide sequence, nucleotide base has been changed. Just one base pair has been changed. And that has tremendous bad effect on our cell. Just one changes had happened. And for that, cell cannot make insulin. And that thing happened when DNA actually replicate. What happened? So this was the sequence on DNA during replication. Because of the mistakes inside cells, so instead of AUG, so U means T, so on DNA, it instead of A, T, sorry, not AT. So if it is AUG, that means on DNA, it has to be TAC. So this was the sequence. Okay. But what happens? Instead of TAC, it made TTC. So mutation actually occurs during the cell replication. So actually my insulin genes should start with TAC, but because of the mutation, I do not have any TAC sequence. Instead, I have TTC sequence. So for the messenger RNA during transcription, cell does not look for any start codon or stop codon. So cell actually will make messenger RNA with AAG codon. And what will happen? Then when it will come near ribosome will will not find out any start codon, and it will not make any protein. So just only one change will bring a tremendous bad effect inside my body. I cannot make insulin, so I will have high diabetes. So that's the mutations. For mutations, the cause of the mutation is called mutagen. Maybe the radiation. If you stand outside under sun for a long time, you can have that mutation from the UV rays. There are so many different types of rays, for that you can have mutation. So many effects of different types of chemicals, high temperature. These are all are the reason of the mutation, and that is called the mutagen. So we have two different types of mutation, spontaneous and induced. The reason behind mutation: spontaneous, random change in the DNA arising from the errors in replication, as I have told that during cell replication, DNA replication. Another one is induced mutation. So because of all these effect, just nitrogenous base pair can be changed, and for that you can get mutation. I gave you just example over here. The original word was beast, B-E-A-S-T. That was the normal word. Substitution. So just one base pair has been substituted. So over here, B has been replaced by F. And the entire meaning of the word has been changed. Now it's not beast anymore, it's feast. Insertion. Okay, so in between B and E, we have a new nitrogenous base R. So it is completely a new word with new meaning to the soul. Deletion, that A has been deleted. The meaning got changed. Inversions. So over here, so we had B-E-A-S-T, that S and T changed their position. That also bring a new meaning to the soul. So these all are the reason for the mutation. So one base pair could be substituted with another. New base pair can be inserted. One base pair can be deleted, or they can change their position, means inversion can happen inside the cell. And all these are the reason for the mutation. So point mutation is called changes to one base in the DNA code, and may involve addition, deletion, substitution. So just single base. We are talking about just one base pair. So it was AUG, methionine. Now it's AAG, means it's lysine. Okay. So over here, I gave you an example. Like for sickle cell anemia, so serine, valine, proline, tyrosine. For sickle cell anemia, so just happen, like let's say this is the sequence: serine, valine, proline, tyrosine. Okay, and if this is the sequence, it will make a regular hemoglobin. But if just one amino acid, if it has been replaced by another one, like let's say valine has been replaced with proline, then it will make abnormal hemoglobin, and that actually give that sickle cell anemia. Just one. Now the point mutation, there are 4 different types. So silent mutation has no effect on the protein sequence. So over here, T, so 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. Okay. So over here, A has been replaced with T. So mutation occurs over here. It was the original sequence, but this is the mutant version. So instead of serotonin, valine, proline, tyrosine, cell will make serotonin, valine, proline, tyrosine. But over here, look that we had mutation. But remember that for one amino acids we have multiple codon, right? So we had mutation, but same amino acid has been produced. So over here, we really do not see any change of amino acid sequence. So it does not have any effect on cell. We had mutation, but no effect. In that case, it is called silent mutation. Missense mutations, over here, just one amino acid has been substituted with something else. We had proline, instead we have thrombin. Okay, so completely different. In that case, we have a strong effect on our cell. Nonsense, if one codon has been replaced by the stop codon. So let's say that amino acid is really long, large amino acids, okay, and messenger RNA. So for insulin, maybe we need, let's say, 30 amino acids to make that insulin. But we had one mutation over here. So maybe this is the number 20 amino acids. If we have mutation on codon number 20, then and if it is a stop codon after mutation, then cell cannot read from 21 to 30. So we are going to lose all these amino acid production, and as a result, instead of insulin, it will make some other types of protein, completely different protein cell is going to make. So in that case, I might have I could get, instead of black hair, I could get red hair for that. Okay, insertion. So shift in the reading frame. So I had sequence A, U, C, G, A. So I have two codon sequence. In between U and C, if I get one code base has been inserted. So cell will start reading this, then cell will start reading this. So before it was CGA, now it is GCG. So completely different amino acid. And in that case, the entire reading frame for the ribosome and tRNA will be changed. In that case, that type of a mutation is called frame shift mutation. So I gave you example. Before mutation, the sentence was the fat cat ate the rat. So this is one codon, 2, 3, 4, 5, 6. 6 codons are there. So when they are in original format, cell can read it. It does have a meaning. The fat cat ate the rat. After mutation, we really cannot read anything. The fat, HCA, TAT, ETH, ERAT. So over here, we have insertion of one new amino acid. So in between C and T, one new nitrogenous base has been inserted. And because of that insertion, the entire reading frame has been changed, and it really does not make any sense. Okay, another one, the fat cat ate the rat. That E nitrogenous base has been deleted, and cell cannot read. And this type of, so entire, so it was not like just, okay, so AAG, so it was proline, instead of proline, it is making AUG, so it is methionine. It's not like that. So over here, just variation of one protein, you can understand changes in the one amino acid can give us sickle cell type of disease. If the entire frame is has changed, then how severe the effects could be. So I gave you few questions like what type of mutation is this? So ATG, it was made, then instead of methionine, it is made LILE. So completely different. So this is an example of point mutation. Now the dog beat the cat. Okay, so instead of cat, it is R. This is also 1 point mutation. But over here, this is the example of frame shift mutation. So I think we are done, and we really do not have time also. Okay, so do you have any question?
No, thank you.
No, thank you.
Thank you.
Okay, so work on that, and
Thank you, ma'am.
we are going to start chapter 12 on Thursday. Okay?
Thank
Okay.
you.
You erased the ink thing again.
Oh. Yeah, because I have class tomorrow, so for that.
Oh, okay.
Also during the recording.
After tomorrow's class, I am going to post that one.
Yeah, but if you were posting the recording of the presentation,
yeah.
then we'll be able to see what I want to be able to see again is the explanation.
Yeah, I'm going to post the recording.
Okay.
Okay?
Okay.
Okay, bye. Good night.
Exam is from the
Marjorie,
13th.
it's your son.
Hello?
Hi.
Say hi,
The-
Lucas.
The exam is
Hola,
from the
guapo.
13th to the 18th, 13th to the 16th. Hello?
Yes.
When is the exam? It's from the 13th to the 16th, right?
I really don't remember. It's inside the syllabus.
Okay, I'll double check.
Yeah, I will follow the syllabus. I cannot remember the exact date at this moment. But whatever the date I gave you, I will follow that date.
Yeah, 14 to 16. That's what you
Okay.
posted.
So then it will remain open from 14 to 16.
All right, thank you.
Okay.
Thank you, ma'am.
Okay, bye. Have a good night.
Thank
as
you
well.
too.
And be safe. Don't go outside.
We won't, ma'am. Thank you. Bye.
Bye.
Good night.
Good night.
Thank you. Bye.
Bye, Marjorie.
Bye.